So welcome. I think we can start this webinar, and I'm asking the assistants to start sharing the slides as we are starting this webinar. I don't see the slides coming up. So, perfect. So, welcome to this webinar, hello, and welcome to the ACM Tech Talk. This is a webcast that is part of the ACM commitment to lifelong learning and professional development, serving a global membership of computing professional students. My name is Michela Taufer. I'm the Dongara Professor in High Performance Computing at the University of Tennessee, Knoxville. I'm also an AAAS Fellow and ACM Distinguished Scientist. I serve as the Vice Chair of the SIG HPC Executive Committee, which is co-sponsoring today's talk. You can find more detail on my background and the resource area in your Zoom console. For those of you who may be unfamiliar with ACM or what it has to offer, it is a little bit of information before the start of this webinar. ACM offers education and professional development resources that increase skill and enhance career opportunity. You can see some of these highlighted in your screen. ACM provides access to the ACM Digital Library, leading publication and global conferences, support for education and research, and the ACM Code of Ethics, which is a collection of principles and guidelines designed to help computing professionals make ethical, responsible decisions. Before we start, we would like to quick mention a few housekeeping items shown on the slide that we are not sharing, unfortunately, but I will remind that to you. If you have questions at any time, please type them in using the Zoom Q&A button. We will organize the question, and Amanda will have an opportunity to answer some of your questions at the end of our presentation. Of course, if we don't have enough time, we will follow up with answer to make sure that all of you have the opportunity to discuss with us, and in particular discuss with Amanda. This section has been recorded and will be archived. You will receive an email notification when it becomes available. And check learning.acm.org for update on this and other upcoming webcasts. At the end of the presentation, you will see a survey open on your screen. Please take a few minutes and let us know what you think about this activity, the tech talks. Give us your feedback, help us to improve that. So now let's move to the exciting part of this tech talk. And today I'm thrilled to present Amanda Randles, and who is here today to present lesson learned from building GitHub. Oops, my goodness, this is not the right title. Developing vascular digital twins to shift from reactive to proactive care. Well, let me tell you a little bit about Amanda. For those of you who do not know her, well, this is a fantastic opportunity for us to learn about her work. And Amanda Randles is the Alfred Winborn Mordecai and Victoria Stover Mordecai Associate Professor of Biomedical Sciences and Biomedical Engineering at Duke University. She has courtesy appointments in mechanical engineering and material science, computer science, and mathematics departments, and is a member of the Duke Cancer Institute. Focusing on the intersection of high-performance computing, machine learning, and personalized modeling, her group is developing new methods to aid in diagnosing and treating diseases ranging from cardiovascular diseases to cancer. She has received the ACM Prize in Computing, the NIH Pioneer Award, the NSF Career Award, and the ACM Grace Hopper Award. She was named to the World Economic Forum Young Scientist list and the MIT Technology Review Awards Top 35 Innovator under the age of 35 list. She is a fellow of the National Academy of Inventor. Prior to graduate school, she worked as a software engineer at IBM on the Blue Gene supercomputer team. Amanda was contributor to over 100 peer-reviewed papers, 121 France US patents, and has about 75 pending patent applications. I'm thrilled to introduce Amanda, and with that, Amanda, please welcome, and without further ado, take it away.
Yeah, thank you so much for the kind introduction, and thank you all so much for joining from, it looks like, all over the world, which is pretty exciting. Especially, I know some of you are up very late to listen to this, and I really appreciate you all joining. I'm really excited to talk to everybody. So we'll get going. So mainly, I've been really, ACM has been a huge part of my career since I've been, an undergrad to graduate student. And I've really enjoyed being a part of the community. They've been a big part of, giving me valuable connections and insights. And I've found it to be one of the more supportive communities. So I'm really excited to get to talk to you all and share a little bit more about some of the research our lab has been doing, and kind of a topic that's really been important to me, where what I'm very passionate about is using large scale computing, advanced algorithms, AI, and really trying to tackle complex biomedical challenges that we wouldn't otherwise be able to address. As a researcher, I'm very passionate, very excited about exploring what we can achieve by using some of the world's biggest supercomputers. I get very excited about using the largest supercomputers we can, trying to find innovative data types, different modalities. How do we integrate across these and accelerate these different breakthroughs? And now my coffee machine is going, so I apologize for the noise in the background. I think in general that this is a really exciting time for computational science in the context of, we just saw the Nobel Prizes come out in this area with computational science being applied, the importance of AI and combining it on that end. And I think this is truly, a testament to how important these fields are and the impact that the computational approaches can have in really tackling these new challenges. So as I was preparing this talk, I think typically when I'm giving a computing talk, I would start pulling together all of our scaling graphs and show the performance data for how we're using these large scale supercomputers. And with this, I really wanted to give you more of a sense of the type of application and the impact that we see these models having, and really get into the role that digital twins can have in healthcare and what some of the opportunities are there. So I really think digital twins are opening up new possibilities in healthcare, enabling us to explore different what if scenarios, giving us more personalized, proactive care. And what I want to do today is kind of give you two different big examples of where we're working in this space and kind of give you a feel for where we're trying to innovate. So with that, I'll jump into some of the description. And when we named the talk, it's really trying to shift to this idea from a reactive approach, which is really the status quo, right? Where we have a patient that comes in, they experience symptoms like they have chest pain, they have weight gain, trouble breathing. It prompts some kind of a visit to the doctor's office or to the hospital. And we want to move away from this reaction to these symptoms to something that's much more proactive. So in a proactive model, we're not waiting for these critical symptoms to occur. Instead, we want to continuously and remotely monitor these patients and be able to predict these health risks before they manifest into a way that could lead to a crisis. So you might wonder how this is different from something like preventative care. And traditional preventative care really focuses on reducing your risk over time. So it has, we include things like screenings or vaccinations or lifestyle change recommendations. Here, what we're really focused on with proactive care is leveraging continuous data to really anticipate and address specific individualized health events before they can escalate. We want to catch these changes early on and enable a timely intervention to keep patients healthier and keep the patients out of the hospital. So what we want to do is identify these risks or these likely issues before they occur. So before you have shortness of breath, before you have chest pain, and say this patient is likely to have issues, they're on the cusp of having these issues, and it may be at a point that the clinician can step in and proactively address that problem and circumvent it. And that's really where we see digital twins starting to play a key role. So we have this idea of, that we have this remote digital twin that is going to be, in sync with that patient, allowing the doctor to have remote, non-invasive, continuous access as patients go about their daily lives to understand what is happening with that patient and help them identify, are they on the cusp of something going wrong? Can we identify that earlier? Can we even use this to identify new different biomarkers, new digital biomarkers, to allow us to identify that potential issue early? So I'll get into a little bit more of like what I actually mean by this. So the first question is, what is a digital twin, and what do we mean in this context as a digital twin? We already know that digital twins are commonplace in other areas of engineering. So you have, companies like GE are building digital twins of jet engines. They're informed by streaming sensors tracking the state of that specific jet engine. And that's the key part here. It's not an idealized model of the jet engine. It's not this, representation of a jet engine. It is genuinely a simulation tied to that specific jet engine, understanding the wear and tear on that engine, where is the current state for that specific engine. And we want to be able to do that for the humans. So we want to be able to, in the same way with the engine, they alert the engineers before the engine fails that, maybe you need to come into the shop, kind of think of like a check engine light. What the goal here is, can we do that for patients? And wouldn't it be great if doctors could have that and say, this is the time you really need to check in with this patient, and maybe time to step in. We think something is going on, and identify those patients earlier before those symptoms occur. So just kind of stepping back and trying to help you guys build some intuition. And this is just a basic example of like how you might think of using a digital twin in this space. So if we looked at something in the very short term, we know currently that we have a lot of guidelines that are based on aggregate population data about how long you should run, how often you should run. It's great to get, moving forward, it's great to get, half an hour of daily exercise, or like these population level statistics. But what we really want to have is a way of identifying what's going to make a tangible difference for that specific person. So say you plan to run for 45 minutes today. How do you decide if you should focus on speed or if you should focus on endurance? It would be really great if you had a digital twin who could know your current state of, how much sleep you had, how much caffeine you've had, what you've eaten today, how stressed you are. And they could go and test, for your specific state on that day, try out an interval, try the steady state, try running sprints, assess the impact for you at that given time, come back, come back, given how tired you are, and tell you what you're going to do is get the best bang for your buck if today you just do a slow and steady state run. We're already seeing the first steps towards this type of guidance through the resilience feature and advice in systems like the Aura application. You're already seeing this kind of, these kind of AI-based analysis and features built into wearable devices, and we're kind of seeing those first steps. What we really want to do is tie it into physics-based models and understand the true physiological state of that patient. And while it's great to figure out, how much you should exercise and how to optimize your exercise routine, what we really want to do is have an impact on how we help with healthcare and how do we mitigate long-term issues for those patients. We want to find ways to identify if we could make a person less susceptible to long-term plaque buildup that might lead to something like atherosclerosis. In the last 20 years, we've gotten really great at characterizing vulnerable arterial states, but we've kind of struggled with trying to convert this into actionable guidelines. Our hope is that digital twins that can capture the blood flow over time might allow us to assess not just if the patient's going to have, their arteries entering a vulnerable state, but what percentage of time they're actually spending in that vulnerable state, and help us to identify what needs to change in order to reduce that. To do that, it'll really allow us to use these digital twins to capture months or even years of time and move the needle in terms of shifting that cardiovascular care from reactive and preventative over to a proactive state. Okay, so kind of getting into what does this actually mean? How do we actually build these digital twins? What my lab is focused on is building vascular digital twins. So we're starting with just the vascular system, focusing just on blood flow modeling as the first step and kind of our proof of concept initial case. For the rest of the talk, I'm going to talk about two key examples. First, looking at cardiovascular disease, which is more of a bulk fluid. How do we translate this for vascular, cardiovascular diseases in this area? And then on the other side, trying to understand how we might use that when you care about individual cells. So the example we're looking at is in cancer cells, but you can imagine when we want to go like a little bit deeper down and not just have the blood flow modeling, but understand how the different red blood cells are interacting, how a cancer cell might move through the body. You can imagine using that for inflammation and understanding white blood cell trajectory, but kind of really getting at that macro scale of blood flow across centimeters or even the meter scale if you're looking at the full body, to really how do we delve down into that cellular cell-cell interaction, more minute area. So we'll spend the rest of the talk kind of splitting up in these two spaces. Starting first with cardiovascular disease. So we'll start playing the video. What you're seeing here is hopefully helping you build that intuition and building a picture in your head of like what I really mean by these vascular flow models, this digital twin. So we're not just talking about a data model. We're really trying to build a 3D representation, a virtual model of the patient's heart that a doctor could monitor and understand what your blood flow is doing as you go about your daily activities. So we want this virtual model of the patient's vasculature that's going to mimic real blood flow over time. And that's what you're seeing here. This is a digital twin of a patient's left coronary artery. I'll come back to this later in the talk. This is actually from 2010, and it has been, it took a lot of compute power to try to get to this kind of computational model. What we're looking at here, and as you're seeing the color change, you're seeing like the velocity changing, and you're seeing the individual red blood cells, but you're seeing the forces and the velocity kind of being higher as you're going through that bifurcation. The amount of time you're spending at these high force regions at the certain locations could indicate the likely disease localization or a location where you're likely going to see progression of the disease. The key point here is it isn't just about the anatomy. It's not just about the heart rate that's going to be predictive of the disease, but it's that synergistic combination. We can't predict where the disease is going to develop purely from the geometry or just from some of the data. We need to augment that information with these physics-based models to understand that 3D blood flow to get this accurate and predictive biomarkers that we can pull out of them. So in order to build them, we need patient-specific images. So we're building this based on CT scans, MRI scans. We have a tool where we can use 2D angiography and build that 3D model. When you're working with data like a CT scan or an MRI, you have a Z-stack of these images. You go through a process called segmentation. What you're seeing here in the middle, you're kind of extracting that geometry. So you're getting a 3D mesh that's going to represent that patient's geometry. So what you're seeing here is a 3D triangulated mesh representing a specific patient's coronary anatomy in this case. We then feed that into a high resolution computational fluid dynamics software we call Harvey. It's named after William Harvey. He's the first scientist that realized that blood circulates through the body. We use large-scale supercomputers. We use cloud computing. We're doing a lot of work on the high-performance computing side, which I'll kind of touch on, and I'm happy to answer any questions on that end. But we're trying to capture that blood flow data in a really fast way, and we're trying to move from being able to capture it over a single heartbeat to doing this over time. So we're linking this data, or we're linking these models to wearable trackers to get continuous information, and that's where it's moving away from just being, a digital shadow or a digital model to truly being a digital twin of the patient, syncing up with their wearable data, driving the simulation and understanding how the blood flow has been changing over time. And that's allowing us to build these longitudinal hemodynamic maps that are giving us that longitudinal view of the, patterns of blood flow over time. And this is kind of giving you an idea, we'll see, of the of that coronary artery and the blood flow changing over time. And we'll kind of skip through and talk about this a little bit further. And this is really like that underlying framework that allows us to create a patient-specific, personalized vascular digital twin. And this is all great. But the next step is then, okay, where are we at now? What's going on in the field? And right now, it's an exciting time to be in the space, especially in the coronary side, because there's such widening acceptance and growing number of like FDA approved tools for these personalized models that are being used to replace invasive techniques. So the canonical example that everyone talks about is like the first pass is looking at this quantity called fractional flow reserve. So we're already seeing tools that are being used in the clinic today where we are replacing invasive guide wires to determine whether a patient needs a stent or doesn't need a stent in their coronary arteries. So the conventional way of doing this is in, like, you can kind of see it in the image, in the image here. There's a guide wire that we place in the coronary artery, and that yellow part is the plaque buildup. So you have a narrowing in your artery right there. And they'll measure invasively with a guide wire the pressure before the narrowing and then the pressure after the narrowing. And it's really just the ratio of those pressures. If it's above or below 0.8, that just determines whether or not you're going to get a stent. There's been a lot of work by many different groups trying to show that these computational models and digital twins can actually use the CT scans, MRI scans, other ways of getting personalized modeling, build the 3D model, and then accurately allow you to measure the pressure in the simulation instead of in the patient, and it measures those it matches those invasive measurements. So we're actually seeing, in the clinic today, the replacement of these guide wires, and conventionally there's 1 million diagnostic catheterizations annually. We're seeing those start to be replaced with these digital twins, and we're showing that these digital twins have the potential to really be as accurate as these invasive tools and allow us to get that that remote monitoring of these characteristics. What I think is even more exciting about this is the potential of these digital twins to uncover even new digital biomarkers. So insights that you couldn't directly measure in the patient, but we can signify enhanced patient stratification. We can identify those patients that maybe should have been treated that we weren't catching ahead of time. So for example, with this FFR case, you have patients that are in the gray zone. So they fall around an FFR, maybe they're not right at 0.8, maybe they're at 0.801 or right in that gray zone on that cusp. And they're missing that stenting threshold, but they really should have been included and should be a part of that. Our models can identify those patients in this range that are at a higher risk of adverse events. So one quantity we showed was the patients in our large trial, the patients who had a higher vorticity and were in the gray zone were much more likely to have a higher adverse event. So this is really laying the groundwork that these markers that you can get out of these complex models might be able to give us these new markers to get new diagnostic metrics to kind of augment what we already know and we're already able to use with the invasive measures, and better stratify these patients to identify patients that wouldn't have been treated otherwise and really should have fallen on that other side, the other side of the threshold. Beyond this, everything you were seeing there was looking at single heartbeats. We're using single heartbeat metrics, we're showing we can match it, and we're really able to use these single heartbeat metrics to diagnose and identify, if patients should get a stent or not. The exciting part that we're at now is we're really seeing this healthcare data revolution. We're witnessing a pivotal shift in healthcare that's really driven by the rise of these wearable sensors that can continuously capture dynamic data as people are going about their daily lives. So, currently, and if you can see, I have my Apple Watch on, I have my Oura Ring on, I have, multiple wearables just trying to track it. I'm sure everyone in the audience is wearing, I would bet most of you are wearing some kind of wearable or even have the step trackers in your phone. In 2022 alone, there were about 115 million registered Apple Watch users. So when you then add in the Fitbit data, the Garmin data, all of the other tools, our phones, the scale of information that we're getting access to is really becoming incredibly staggering. So traditionally, doctors relied on data that they were collecting in the office with a single snapshot in time. And I'm sure all of you have had this experience of, something is going wrong, you don't feel well, you're having some kind of reaction, you go to the doctor's office and everything seems all better and, it's not being measured any longer. If we have access to this wearable continuous data, we actually can track it and have a record of that event, and we have access to even more dynamic information that might be more insightful. So this unprecedented amount of data is giving finally the need for these flow simulations to actually go beyond a single heartbeat. We didn't need to do that before because we had no reason to act we didn't have the dynamic data of what was your heart rate across the whole day. Now that we have access to it, we have a newfound need to actually drive these flow simulations, not just for a couple heartbeats, but can we actually do it for a week or a month or see, what is your actual physiological state over longer periods of time. So it's been really exciting on our end of there's finally a need to really go past it But then we have this problem that it's still a huge computational challenge. So coming back to this video that you guys watched before, these flow models are really computationally intensive. Again, like this one's from 2010, so we've seen a lot of advancements, but I think this just gives you kind of a sense of understanding. This video that we showed was run on one of the world's largest supercomputers in 2010, and it took about 6 hours to model just one heartbeat on the entirety of one of the biggest supercomputers at Argonne National Lab in 2010. That's not going to be as useful if we want to translate this to, moving from a single heartbeat to the millions of heartbeats you would have over just even a couple of weeks. For digital twins to really be clinically useful, they need to be fast, and they need to be fast enough to provide real-time insights. It's a lot like weather modeling, so a good example to kind of build this intuition. We had a few weeks ago in the US and North Carolina, we had a large hurricane. It really, kind of reminded us all of that importance of the timely forecasting. It was critical to know where the hurricane was going to hit, like where it was going to hit on land, how do we prepare, how bad was it going to hit? And while I was really lucky to be far enough away from the affected areas in North Carolina, it was, people were really relying on these simulations to know where it's going to hit and give them a heads up to be able to prepare. Imagine we have the most, accurate hurricane simulation that could pinpoint exactly the point of landfall, exactly how hard, like what the strength is going to be based on the sensor data. But if that simulation would take 3 weeks to complete and the hurricane's going to hit, in 3 days, that's going to be far too late and it's not going to be helpful. The same idea applies to health. So imagine you have a model that could take today's physiological data and everything you're getting from your wearables, and it could really act like this check engine light for your body. It could predict with precision the risk that you have of a heart attack, your likelihood of a heart attack, next week, or what's going to happen. But if it takes you, if it takes 6 months for the simulation to run, or it takes a year for that simulation to run, but the heart attack is going to happen, a month from now, it's a little less useful to have that information. So what we really need to do is find a way to get these simulations to run quickly and be actionable. And the problem here is even moving from 2010 to 2024, where we are with simulation and where we are with the chips, simply waiting for these faster processors isn't enough. When we began looking at this issue, the longest running 3D blood flow simulations that had ever been run had only lasted about 30 heartbeats, which gives you like a little less than 30 seconds of time. And our goal was really to get to, we love to have a week of time, a month of time. Ideally in the grand challenge, you'd want like years of time to be able to track patients and what they're doing. So to make digital twins really practical, we had to drastically reduce the simulation time without compromising our accuracy. And that's where, luckily I get to work with a really amazing team here at Duke University. This team was led by Cyrus, and you see his picture in the top. He's a fantastic graduate student that was willing to kind of go along with me and try something a little out of the box and crazier. And together our team was able to tackle 2 of these big challenges addressing this problem. So not only did we develop a way to drive these simulations using data from wearable devices, but we also created this new parallel in time approach that we called the Longitudinal Hemodynamic Mapping Framework, LHMF, which you're kind of seeing here. And I don't have time to go into all the technical details for it, but again, I'd love to answer questions and go into this more at the end if you're interested. But the idea here is we're taking a parallel in time approach. We're breaking down basically the causality of the model, taking something that should be dependent on itself, turning it into a more embarrassingly parallel problem, extending the time domain that we can model significantly. And then we tested it by dedicating the majority of our local cluster here at Duke for about 4 months, explicitly modeled a few hundred heartbeats, and then we compared the results that we produced with LHMF to, with that, we were just using some cloud resources and a few nodes in the cloud, and compared that to the brute force model of where we shut down the cluster for several months, and showed that we were actually able to get the high accuracy. So what you're seeing here in the black is the explicit model, and that's what the black dots are here. We have the LHMF in the green, and we really were able to have less than 1% error when you're comparing the 2 results. So we really showed you are actually able to do this in a much more computationally tractable way. And doing that allowed us to move far beyond the conventional, what had been done before of only 30 heartbeats. And we were able to capture to date about 4.5 million heartbeats for one patient, which is about 6 weeks of time. We're able to capture this data and drive it from this wearable device, drive this blood flow simulation. That's kind of what you're seeing. If you watch it very carefully, this is the wall shear stress map. Wall shear stress is associated with the development, progression of cardiovascular disease. You can kind of see it flickering as it moves along. What we're able to do is look at different regions of the body and see, how long are they exposed to these different, adverse levels of stress, and not just are they exposed to it, but how much time are they spending in that adverse state. And then previously, we would have only been able to compare, we would have a representative, and I'll show you this in a second, we'll have a representative resting heartbeat or a representative exercise heartbeat, but we wouldn't be able to get that dynamic shift across the entire time. And those results are really and truly like statistically different. So what you're seeing at the top is the result over a long period of the LHM, and how it would compare to our representative canonical, here's a representative resting heartbeat, here's a representative exercise heartbeat. And you really do, you're missing a lot of data by taking these single snapshots in time. And it's incredibly important to be able to monitor how the flow is changing and capture it in that way. So the idea of this LHM using this LHMF framework, it's allowing us to really shift away from just capturing a couple heartbeats to really capturing longitudinal flow with a large number, longitudinal flow over a large number of heartbeats. So I'm really excited about the idea of what we can do with the vascular digital twin. This is exciting for us because it really is like the first time we're able to link up personalized cardiovascular flow models with real world activity. So we're getting the real world data from these wearable devices, capturing how your blood flow is really changing over time. And this level of detail is setting the stage for more key advancements in how we understand cardiovascular disease development, vascular disease, other areas. We're working with clinicians applying this in the carotid. We're looking at heart failure. We're looking at a wide range of diseases to really help us try to identify new biomarkers and allow us to predict the risk, tailor specific interventions, and really optimize our treatments with unprecedented precision that we've ever been able to do before. With these computational advancements and combining them with these breakthroughs in wearable technology, we really have a unique opportunity to continuously and automatically screen patients for these early signs of disease as they go about their daily activities. So it's one of the key pieces of we can actually, track as they're going through, while they're reading, while they're sitting on the subway, whatever it is they're doing in those activities. We have a true view of what they're actually doing versus just that small snapshot of what you would conventionally get in the clinician's office. As I mentioned, we are collaborating with doctors and across a variety of specialties here at Duke, and some in other hospitals around the country to try to capture the real patient data as they engage in these everyday activities. And we're integrating this wearable data with these vascular digital twins, giving us this real-time remote monitoring of the health status. And our hope is to really pave the way for this truly personalized real-time healthcare. The other piece of this that I think is really exciting is, aside from just real-time remote monitoring, we're able to move beyond the digital twin to think about more forecasting. So by using, coupling it with AI surrogates, coupling with more optimized high performance computing side, by making this more computationally tractable, we can also then say, what are different hypothetical paths? What if this person has a high level of exercise over the next 6 months? What do you think could possibly happen? What happens if they were more sedentary? What if, we can do virtual stent placement. And what if we place, stent 1 versus stent 2, and then they have these different activity levels? We can forecast what are the different options of what do you think could happen for that patient before the doctor ever sets foot in the operating room. So we can not only give personalized guidance of, it would be, maybe in your case, you really do need to exercise more, or exercise in the morning versus the afternoon, or what, how can we add that? But also for that specific patient, how do we expect them to behave with different treatment options? So if they gave stent 1 versus stent 2, what do we think that's going to translate to over the next 6 months? And can that help us better inform, can we fight against things like in-stent restenosis, where you have the narrowing come back, and really reduce those options? So we're hoping to use these prospective models to guide treatment and really require, sampling these hypothetical scenarios and allow us to better inform treatment and have this forecasting potential as well. So I'll pause and like move then. I also want to spend a few minutes before I wrap up and just take the last bit of it, telling you a little bit about our digital twins on the cellular side. So everything I've talked about so far is at, you know, a large macro scale, thinking of the fluid as a bulk fluid and really looking at, you know, the effect in cardiovascular disease. We're also really interested in, if you have to go down to the scale, like what happens at the cellular level. Can we get digital twins that are going to take into account individual, you know, your hematocrit level, like the density of red blood cells, if you have anemia, if you know, how do we take into account these different blood disorders? How do we look at cancer and try to understand how a cancer cell is moving through the body? What is it about that cancer cell that's moving from a primary tumor through the body that's going to make it more or less likely to create a metastatic site somewhere farther away? So trying to create these like cell specific models to really inform our understanding of the mechanisms driving the progression and localization of these diseases. In order to do this, we are building a multi-scale, multi-physics model where, you know, it's starting with Harvey, which is our underlying fluid simulation, which is a high-performance computing model that is, you know, parallelized with OpenMP, MPI, ported to all the different programming models like HIP, SICL, and all that, which again, if you want to talk more about at the end, we can talk more about. We rely on the Lattice-Boltzmann method, which is an alternative for the Navier-Stokes. We're using the immersed boundary structure for the fluid structure interaction, and we have a finite element model of our cells. All to say, like, it is, you know, when you're modeling these cells and you're looking at these simulations, you have potentially hundreds of millions of red blood cells that are going to be interacting with each other. Those red blood cells are deformable. They're interacting with the fluid. They're moving through the fluid in a complex geometry. And you not only want to have these like cell-cell interactions, we also care about the adhesion. So if the cancer cell is moving through the vasculature, why is it adhering to different spots? What is causing that adhesion? We want those individual ligand receptor interactions. So we have this truly multi-scale model where we want to capture all the way from, you know, the system scale of the full body down to that adhesion level, which as people interested in the ACM, I'm sure you can fully appreciate why that is a huge computational challenge. So the way we started looking at this, the first step is just looking at a single cell. The first, like, you know, the critical first step is being able to actually accurately model these cells. So we did a lot of work. This led by like Peter that you're seeing on one side, Marianne on the left, former postdocs in the lab that really helped us set up our cell model and ensure that we are accurately getting the properties of these cells. So at the top, we're showing the red blood cells. We mimicked micropipette aspiration. We mimicked optical tweezer experiments and ensured that we were building that individual red blood cell model to match experimental data. So we truly are getting the right model of your red blood cell. On the cancer side, it was very important then that you're getting the cancer cell correct. And in that case, we want to be able to tune your cancer cell model to match different cell lines and match different cancer cells. So here we're just focusing on, you know, starting out and making sure you have one cell correct, then scaling up to many different cells. We worked with Scott Manalis's team up at MIT where they have this device where the cancer cell is going through this constriction. And we tuned our cell model and built the cell model so it could be tuned to different cancer cells. So what you're seeing here are two different cell lines. We have T-METs and the L1210, and showing that we can actually capture the nuances of the different cell models with our computational tool. And we're looking at the passage time through this constriction. So we're trying to make sure we're mimicking those experimental data. Once we have the single cells in place, we're then trying to scale up to a computational model that can go from the single cell to tens of thousands to hundreds of millions of red blood cells and really understand it on a larger scale. So we really start, you know, step one, make sure you know one is correct, and then build up from there. In applying it to cancer, we worked with the team at Lawrence Livermore National Lab that was able to create this this hydrogel and this like endothelialized printed channel where we were creating, we had an endothelialized channel that you're seeing here. It's about 18 millimeters across. We then were able to put circulating tumor cells through this device. And what you're seeing in bright red is where those cells are really adhering and sticking to that channel. So we were able to then really in fine detail, try to understand what is it about the channel that is causing these cells to stick in different locations. The interesting piece here, and you can kind of tell this is, you know, it was the STL file, like the mesh file that was provided, was meant to be just a double bifurcation channel that is mirrored. But you see, like when you go through the printing process, it's, you know, it's not exactly like there are, like, it's not perfect. It's a little bit different than what you might expect. And that's where you see like this, it's a little bit narrower here than it is up there. But the interesting piece there is that that led to different behavior. So we saw more adhesion in this region at this bifurcation than we did up here in the real experiment. And it gave us a lot of data to really look at and try to understand what is it about those geometries? What is it about the fluid flow in that region? What is it about the cancer cells in that region? Why are they adhering in those, why are they adhering there versus other points? And how do we really start trying to understand that? However, one of the big problems was at this time, and this was in about 2020, so it wasn't that long ago, this might seem like a really simple geometry, but trying to model all of that at cellular scale or resolution on the large systems was just not possible to capture all of the cells you might put in there and getting that level of resolution. So we were able to understand the wall shear stress in that region, but we had to do a fluid only simulation at that stage. We then followed up to do an in-depth understanding of trying to see how does the cell interact with those bifurcations, and kind of just did a zoomed in approach with what we could do that was computationally tractable in just an idealized region looking at the bifurcations. So that's kind of where we were in about 2020. In 2022, Daniel, who you'll see on the top here, was a former grad student who's also been very lucky of having fantastic graduate students, where you can kind of see here what he put in is this adhesion, this individual ligand receptor adhesion model. So the little yellow pieces you're seeing at the bottom are individual bonds that are being mimicked and formed. From the computational end, what that means is we have to actually capture that sub-micron level. The resolution of that model has to be that fine to be able to get those individual ligand receptor pairings. The interesting thing in this video, and I'll start trying to go faster so we have time for questions, if you include the adhesion, it changes the trajectory of that cell. So what this is showing is it's really important to include behavior like adhesion. We were then able to parallelize this further, and he was able to get it to the point where he can actually capture adhesive cells on the order of about 570 microns across this device. Keep in mind that's still a far cry from this 18 millimeters we would like to get to. Show the quick video of just kind of getting this idea of we can have multiple cells going through, and we're making use of these large scale supercomputers, capturing these deformable cells, moving through and interacting. But it's still not where we want it. So the quick version that I want to just introduce you to this idea of our moving window. It's our adaptive refinement model. We call it the adaptive physics refinement approach. The interesting piece here, if you're going to brute force it, just use the world's biggest supercomputer and put as many red blood cells as you possibly can. So at this time we were using Summit at Oak Ridge National Lab. We used the entire thing, and we could model 550 million red blood cells, which is huge and sounds like a lot of cells to model and is really exciting, but it's really only about a 3.1 millimeter tiny little cylinder. So that's only a small part of the vasculature and really not capturing the domain that you would want to get to. Even in that device that was 18 millimeters across, you can see we're not really getting there with a 3.1 millimeter cube. So what we introduced was this idea of a moving window. So it's basically you have a high resolution window around your cell of interest. So for us, that's a cancer cell. Inside the window, you have all of the different red blood cells interacting. Outside the window, it's a bulk fluid, and then it's going to move through. So that way, the hypothesis here is the orientation of the red blood cell when the cancer cell hits it is going to affect the location of where that red blood cell is going to move. But the orientation of the specific red blood cell 5 centimeters away from the cancer cell is not really going to affect its movement. You really just need to capture the bulk fluid properties. So we have this hypothesis that by having all of the cells explicitly modeled in a small region around the cell of interest is going to allow us to capture the behavior as we move along. It's kind of just a quick animation made by Liam at Livermore of kind of showing, basically you have this box of cells that's going to move around and show as it goes through. We'll go through that. The key part here is that allows you to drastically cut down the amount of memory that you need. So the high resolution bit is only going to be in the box. You have bulk fluid in the rest. It's at a coarser resolution, and it's really, so here's our explicit fluid structure interaction model took 22 terabytes. Using the APR model, we could get it down to 216 gigabytes. So it really makes it possible to do this. I'll show quickly just the validation side of here's your explicit flow solver where you're putting all of those little tiny individual red blood cells. We mimic it with the cell, with this APR window box. And basically the takeaway here is we get the same trajectory of the cell as it's moving through. We used an expanded channel here, which is important because it's showing that you see the margination of that cell as it goes through. if you imagine that window and what it's seeing from the window doesn't know here that, just if you were just modeling only the window, it wouldn't know that it had gone through an expansion, so you wouldn't see that margination. But because of the coupling setup within the APR system, you're coupling it to that larger the larger bulk fluid domain outside, which is giving it that sense of the overall geometry, and it knows that it should be marginating, it's hitting the overall flow, but then it is still capturing that individual cell interactions. So with that working, we're able to kind of show that you can get the moving window and actually start tracking the movement of cells across multiple millimeters on a much larger scale than we would ever have been able to do before. Remember, we were able to get all the way from here to here in about 96 node hours. Keep in mind that just doing that little tiny, like 3.1 millimeter cube took all of Summit, which was just, would have been 4,600 nodes all at that time. So it completely changes the kinds of models, the kinds of questions we can ask. So I want to just end on the now we're adding in, and Aristotle was able to present this last week at BMES. It's really exciting direction of the keys now added adhesion into the moving window. You can kind of see the cell adhering as it moves through. So now it's taking it even a step further. We can actually capture adhesion over a long period, long distances, and really start to assess the effect of these models, the effect of these properties like adhesion, different cell properties, as it's moving through real complex geometries and getting to move away from these idealized systems. So I'll stop there and just kind of wrap up with there's still huge computational challenges. These large scale simulations that you see in these digital twins, whether they're vascular or cellular, the computational complexity is huge, and it really, there's a lot of places for people to innovate and to get involved in these areas. We care a lot about how do you visualize the data. We're generating petabytes and petabytes of data. Visualization, data handling in itself is a huge challenge for us. As we start working with all these different supercomputers, we know that it's not just, CUDA-based systems anymore. We have to prepare for sickle models and HIP and HOCOS and all of these different programming models. We're developing machine learning surrogates. We're trying to have reduced order models. There are a lot of challenges that are kind of open as we try to get these digital twins more useful and more, adapted in the space. And with that, I'll wrap up on this of I've really talked about vascular digital twins today, cellular digital twins. Our goal is to be able to integrate, diverse patient data, genetics, immunology, neurology, kind of, use this as a proof of concept so we could have these more comprehensive predictive models in the future that are going to adapt as new data comes available. And you can kind of imagine future scenarios where we're going to incorporate data from implantable devices tracking, real-time blood chemistry, cellular interactions, joint stress, and really see these kind of become much more pervasive. And I think digital twins are much more, this is really just the beginning. It's a powerful area. And I'll stop there and say thank you very much for all of your attention. I'm happy to take questions, and thank you to my team and everyone who made all this work possible.
Thank you, Amanda, for this fantastic presentation. And we have now time for question and answer. Several of you have been submitting fantastic questions. I tried to gather some of them together so that in the next 10 minutes, Amanda will have the time to tell us more about your question. But I remind you that we are recording your question and we will review them and perhaps follow up. So one question that came from several attendees is whether you can elaborate the difference in definition of digital twins versus traditional simulation, and also revisit in terms of what is the difference, what is the unique challenges that are present in digital twins, but have been overcome in traditional simulation. On the other hand, some people are asking also what distinguish digital twins from traditional simulation that are based on patient-specific model for human anatomy, and they're referring to the fact of integrating diverse multifactorial data like genetics, hormones, and physiological factor into a model that is not a digital twin. So if you can help us understand a little bit these differences.
Yeah, I think that's a really good question. And definitely makes sense if people are like, it really depends who you ask. So that you'll hear a lot of different definitions of digital twin right now. It's being used in a lot of different ways. The way we're using it, and I would argue, is I think, we have these personalized patient-specific models that we've been working with and for the last decade or two, right? Where we're creating, you take a CT scan, you segment the data, you get the geometry, and you're running a patient-specific blood flow simulation. And you might even be tuning that blood flow simulation with data from the electronic health record, but it is a standalone simulation that is not dynamic and not, tied to the patient beyond, like you take a static snapshot of that patient, what their data is, and you run a model. And I think that is, in some cases, we're seeing that being referred to as a digital twin. To me, I would say like that's still like that's a patient-specific model. It's our personalized flow simulations. It is, critical and important, but kind of not really bridging that digital twin side. I think when it really it starts to get to the digital twin side, some people might refer to this, like I've seen more as the digital shadow side of like, you need to start having that connection to the patient. So we want to have some kind of dynamic feedback back to the patient. So for us, we're seeing it as like the shift to where you're taking the data, where you're driving the simulation from the wearables and having that continuous sensor data from the patient or from the user that is having that model stay in lockstep with the patient and having it stay, you're having that feedback loop. So it's the dynamic nature and the feedback loop that's really taking it from just being a personalized simulation to something that's closer to the digital twin. You get even closer depending where you're going with, if you have the virtual treatment and that side of things. But I think it's like we're really seeing the digital twins and the use of it as more like once there's a dynamic connection and feedback loop with the physical entity. So you have like your digital asset that is being, constantly, dynamically, continuously informed in a remote way by the user and not just like the static snapshot. But it's a great question because it's definitely like there are a lot of examples, right? Like there are a lot of definitions right now. And when I'm talking about it, I mean like the personalized flow model is a static snapshot. The connection with the wearables is really what's shifting us into the digital twin space.
Thank you, Amanda. Several of our attendees raised the concern about data privacy and protection. Others raised the question about the data security. So from one hand is, how do I ensure patient privacy and protect sensitive patient data? But on the other hand is also, how do I ensure the integrity of the data and model that are not falsified? What is your answer to this question?
Yeah, I think you could have entire lectures on both of those. They're really interesting, and it's complicated, and we're spending a lot of our time talking about how do we handle that now. I think a lot of like even at Duke, when we're trying to figure out how to set this up, more of my meetings and more of them are starting to be about, where do we store the data? How do we store it accurately? Like even for the research side, like it's a constant challenge, it's a constant battle, and something we really need to make sure we're doing accurately. I come back to the privacy in a second. The other piece is also just, it is amazing that so many people are wearing wearables, and this is getting to be so pervasive, but it also changes the game. And, false positives and false negatives, the numbers are just, astronomically different. If you have 1 million people or 1 billion people all wearing these wearables, we suddenly, you don't want to overwhelm the health system by having a 1% error, and suddenly, thousands of people are going to the hospital saying, I think I'm having a heart attack because my watch told me it is. And, it was just a false positive. So it's like we need to be really, really careful of like what information we're giving, how to do it in the right way, how to make it measured and make sure it's being accurately interpreted, and what do we like, how do we handle that alongside the fact that you have, you don't want, like if you think about the watch, you don't want like my toddler to pick up the watch, put the watch on, and suddenly like it takes a measurement and thinks that, this is completely wrong data. Or if, a lot of what we're doing, you're dealing with all the missing data problems of, what happens if someone's, if you're just charging it or whatever you're doing, it's suddenly a lot more complex where we don't know, you have to make sure the patient is wearing it. You have to make sure that patient is wearing it. You have like, how do you handle the missing data? And then a lot of the work that we're doing is based on, like the CT images or the image, like, the image-based geometries that can change over time. So it's great that we want to get to the point where we're looking over time, but if you have plaque buildup or you have, that stenosis come back, at what point is your image no longer valid, or when do you know that you have to go in for new imaging? So I think a couple of the points, like one of the things we keep pushing is like you need to be measuring two items with the wearable. So one is the metric of interest, but something else is like what you're calibrating for and ensuring that you're still measuring the right person, that the image is still valid, that the model, like something from a calibration standpoint to make sure that the model you're using is still valid in this case. So we have that side, and then security is just like there's at this stage, a lot of what we're working on is just trying to set up the discovery phase of like, are there new digital biomarkers that could help us identify heart failure earlier, could help us, predict in-stent restenosis, which is more on the clinical trial stage and securing data in that versus like all of the problems that come with like what do we do when we actually want to deploy it and translate it. So there's different challenges at each stage, but it is the way we're handling it here, we're working very closely with the health system and making sure we're developing the data fabric and the way that we're storing the way we're setting everything up for the studies. We're kind of doing it hand in hand with the side from the hospital. At Duke, we're really lucky where the hospital is literally across the street from where I'm sitting. So we're really trying to work very closely, but it's requiring these interdisciplinary meetings and really, we can't just as computer scientists or physicists go and create the tool that we think is going to be perfect and just try to apply it to all the health data. Like it has to be done directly with the clinicians, and even just anyway, with all these digital twins, like we can't just hand them, they have to be involved with like, how can you actually deploy it? How would it be usable? How can they actually like make use of these tools in the clinic? So we're working really closely, and I don't have a good answer of how do we handle the privacy. It's an ongoing, it's something we have to be considering at each stage.
We are almost at the end, and I want to cover two questions that hopefully will be a little bit shorter. The first one is, how far are we from real-time digital twins? Consider the computational and storage demands and the resources, the computing resources, cyber infrastructure that are available today. Because the tools you're showing us are fantastic, but how far are we from having these tools accessible, real-time support for humans?
I think we're actually, it depends, if you're constraining it to the vascular flow modeling and trying to do like, those studies that we were showing, I think it's within the next few years. Like everything we've been doing, like with the LHMF, we're showing deployment in the clouds. We're trying to figure out how, we're trying to create this so you don't require the large scale supercomputer to be able to do it, and really introduce new algorithms to allow you to break it down and use more commodity hardware that's accessible to anyone in that side. So I think it's much nearer term, but only for these specific use cases. It's not going to be the immunology tied in with all of these really complex digital twins that we would love to have. I think we're still a few more years out.
And a last question, because we are running at the end of time. What are the entry points? Many of our attendees are very fascinated, would like to know, where do they start if they want to be part of this research domain and contribute?
I think there's so many open, like there's so many open questions, and no matter what you're really interested in, like if you care about visualization or if you're interested in the AI side, like there's a lot of opportunity. There's so much opportunity in so many different areas to jump into. I think it's like kind of figure out which part you're most passionate about, what questions are you interested in, because there are AI questions, there are like visualization domain questions, there's HPC optimization questions, and there are people like we're working with teams at national labs, we're working with teams at the hospital. I think it's really like figuring out which domain you're most interested in, and then I think there are digital twin groups across, like they're rising. We're seeing a lot more kind of, if you talk to the national labs, if you talk to different industry, like people in industry, reach out to me if you're interested in getting involved in anything. I'm happy to talk to people. But there's a lot of opportunity in each of these little spaces of just trying to get into like, how do we access this data? What can we do with the data? And I would say like, there's a lot of open.
I feel, Amanda, that we need more time to address all this interesting question and discuss more about this fascinating topic. I'm afraid we're running out of time, and I want to thank again, Amanda, for this fantastic presentation and insightful answer. This talk was recorded and will be available online in a few days in learning.acm.org. You can find announcement on upcoming talks and other ACM activity on learning.acm.org and acm.org Also, please fill our quick survey. We want to learn from you. We want to hear who you would like to have as the next speaker. On behalf of ACM, the SIG-HPC, Amanda Randles, and myself, Michela Taufer, thank you again for joining us today. I hope you have enjoyed this discussion, and I look forward to seeing you in next webinar. This concludes the talk. Thank you, everyone. Bye-bye.
Thank you.